Hello, my name is Chandra Bellasio, and in this video I'm showing you the general features of this Excel fitting tool version 33, which will derive a comprehensive set of photosynthetic parameters from gas exchange data derived under ambient and low oxygen. Here we are in the first spreadsheet, which is called row, and in this spreadsheet raw data are inserted and limitations are assigned to each data point. The light curves and the ACI curves are automatically plotted below the table. So to start this example, I will get some data from the tobacco dataset I included in the paper. I just get the first dataset, 30 July. Copy all the data and paste them in the EFT. So these are our light curves and these are our ACI curves. The color code is maintained throughout the spreadsheet. Blue is always given to low oxygen data points, whereas brown is always given to ambient oxygen data. Alongside each data point is assigned a limitation. One is assigned to the initial data points, in this case of the light curve. Two is assigned to the light limited data points, in this example a light intensity up to 200 micromoles, and three is assigned to the remainder of the data point. The same limitations are assigned under low oxygen. Similarly, for ACI curves, one is assigned to the initial part of the ACI curve, two is assigned to the Rubisco limited part of the ACI curve, generally obtained with subambient CO2 concentration, and three is assigned to the ribulose regeneration limited part of the curve, which is generally obtained under enriched CO2 concentration. We will see in each sheet how these data points are automatically recalled. Here I just want to mention that data points can be excluded from further analysis by inputting a different limitation, for example 2.5 in the middle and 4 at the end. Sheet number 2.3, where a straight line is fitted to the initial part of the light curves obtained under ambient and low oxygen. The data used are those data points assigned the limitation 1, so the initial part of the light curves. The parameters derived are respiration in the light under ambient and low oxygen, and the initial yield of photosystem 2, which is extrapolated to 0 light intensity. If the plots are linear, no action is needed. I want to exemplify how changing the limitation will automatically change the plots in here. So for instance, if I go back on the raw data and assign one more data point to the limitation 1, this will be automatically plotted here and our regression will be 1 point longer. The regression to obtain the yield of photosystem 2 under 0 light intensity is not plotted here, but will appear in additional features, and I will describe this in a separate video. Sheet 4a models the light dependence of gross assimilation. Gross assimilation is calculated by summing assimilation, which is measured, plus respiration in the light, which was derived in sheet 2. The model function is empirical and is a non-rectangular hyperbola. This hyperbola has an initial slope which is YCO2LL, that is the maximum quantum yield for CO2 fixation, and an horizontal asymptote which is GA sat, the saturated rate of gross assimilation. The hyperbola is also described by a curvature, which in this sheet we call M. To facilitate the interpretation of the curvature, the sheet also automatically calculates ppfd 50, which is the light intensity which half saturates gross assimilation. Further, the sheet calculates the light compensation point, which is the light at which net assimilation is 0. This sheet automatically recalls the whole light curve, so all those data points assigned limitation 1, 2, 3. Further, it is possible to specify what respiration to use. For instance, I could add a number, or I could say ambient to recall respiration in the light derived under ambient oxygen. To use this spreadsheet, the solver pack needs to be activated. If the solver pack is not activated, it can be activated by going in File, Options, Add-ins, and here Manage Excel add-ins. A window will pop out where solver can be ticked and activated. To fit the curve, I open solver. Solver is a package which allows to fit one cell by changing other variable cells. In this specific case, my objective is the sum of squared residual in cell M5. It is important that the dollar sign always appears. We want solver to minimize the sum of square residuals, that is, the distance between the fitted function and the data points has to be minimized. The variable cells are M2 to M4, which are the parameters I previously described: the initial quantum yield for CO2 fixation, saturated rate of gross assimilation, and the curvature. Further, I added some constraints here. The initial quantum yield of CO2 fixation is constrained to physiological values, meaning smaller than 0.2 and greater than 0.001. Gross assimilation, the saturated rate of gross assimilation needs to be greater than the minimum rate of gross assimilation, and the curvature needs to be smaller than 1 and greater than 0. If I click Solve, Excel will find the values which minimize the square residuals. And now the plot shows that the function has been fitted. Now these three values describe our light curve. Sheet 4b is very similar to sheet 4a. The modeled function is the same, is a non-rectangular hyperbola fitted to the light response curve. The difference here is that the data are those derived under low oxygen. Similarly, we can use respiration in the light derived under low oxygen. The way parameters are fitted is exactly the same, so I will just fit them. Here I would like to comment on this additional possibility which allows to log transform residuals. The residuals are the mathematical distance between the data point and the fitted function. By log transforming the residuals, the proportionality between the residual and, in this case, ppfd is reduced. As a result, the initial part of the curve has a better fit than the final part of the curve. So we can try this feature typing y here and solving again. Now the fitted curve is more distant in the final part and has a tighter fit in the initial part. Sheet 5a fits a non-rectangular hyperbola analogous to that we have seen in sheet 4 to the ACI curve. In this case, to the ambient oxygen ACI curve. The data used are all data points of the ACI curve. That is, all data points assigned limitation 1, 2, and 3. For the fitting procedure, respiration in the light is not required. It is required only to get an estimate of Ci star, which we will see later. The parameters derived are carboxylating efficiency, which is the initial slope of the ACI curve, ASAT, which is the horizontal asymptote of the ACI curve, meaning the CO2 saturated assimilation rate, omega, which is the curvature of the hyperbola, and gamma, which is the compensation point evaluated on CI basis. To facilitate the physiological interpretation of omega, the sheet calculates CI50, which is the CO2 concentration which half saturates assimilation. Similarly to what we have seen in sheet 4, the fitting procedure consists in finding the parameter values which minimizes the difference between the modeled function and the measured data points. This difference is computed as sum of squared residuals. So I now open Solver. I set the objective cell as M6, which is the sum of squared residuals. I specify minimized, and then as variable cells I add these four cells: the carboxylating efficiency, saturated rate of assimilation, curvature, and compensation point. To speed up the fitting procedure, I also added some constraint. ASAT should be higher than the minimum rate of assimilation. The curvature is smaller than 1 and greater than 0, and the compensation point is non-negative. I can now click solve. And the curve has been fitted. The fitted function is used also to calculate Ci star. Ci star is the substomatal CO2 concentration in which gross assimilation is 0. By specifying a value for respiration in the light, in this case ambient, but we can also type any number, Ci star is calculated from the fitted curve. Sheet 5b works exactly the same as sheet 5a. The difference is that here the ACI curve measured under low oxygen is plotted and modeled. By solving those four parameters, we can now get the parameters which will describe our ACI curve under low oxygen. Similarly, Ci star is calculated from respiration in the light derived under low oxygen. Sheet 6a7 includes the procedure of Yin to derive two important light conversion coefficients which are used later in sheet A8, 10 and 12 to derive J. These coefficients are called S and YJLL. S lumps the fraction of ppfd harvested by photosystem 2 together with some of difficult to measure parameters, which are the engagement of cyclic electron flow and the electron partitioning to alternative sinks. And YJLL is the initial or maximum light conversion efficiency into electron transport rate J. The data used here is the initial part of the fluorescence light curve measured under low oxygen. So data points which were assigned the limitation 1 are automatically transferred here. The derivation of these two parameters is completely automatic, so the only thing needs to be done is to check that the plot is linear. Similarly to what we have seen before, the limitations can be adjusted to add or remove data points. Sheet 6b7 calculates two important light conversion coefficients according to the calibration of Valentini. These are B and alpha beta. Alpha beta represents the light partitioning to photosystem 2, and B represents the fraction of the yield of photosystem 2 which is not used by photosynthesis and the photorespiration cycle. YJLL is the initial and maximal quantum yield for electron transport and will be used later in sheet 8-9. In G3 we can specify whether to include both the light and the ACI curve, or just the light curve, or just the ACI curve. So if we type L, for example, just the light curve is plotted. If we type A, just the ACI curve is plotted. And of course, if we type both, the two curves are plotted. So another possibility is to limit to the light limited data. So if we say yes the sheet will copy only the light curve data points which were assigned limitation 1 and 2, and the ACI curve data points which were assigned limitation 3. If the plot is linear, no action needs to be taken in this sheet. All the parameters are automatically calculated. Sheet 8.9 contains two logical steps. First, the sheet calculates the rate of electron transport J. Then, it models the light dependence of J using a non-rectangular hyperbola. The data used is the whole light curve measured under ambient oxygen. So all data points assign the limitation 1, 2, 3. J and can be calculated with three different approaches: the Yin, Valentini or Bellasio calibration, and these can be specified in cell H2. In this case, Yin is specified. The fitting procedure and the fitted function is very similar to what we have seen in sheet 4 and 5. The hyperbola has an initial slope, a curvature, and an horizontal asymptote. The initial slope is YJLL, the initial quantum yield for electron transport. This is not fitted concurrently in this sheet, but was previously derived with the Yin and the Valentini calibration in sheet 6A and 6B. The horizontal asymptote of the curve is J sat and is fitted here, and theta is the curvature of the fitted curve. To facilitate the interpretation of theta, the sheet calculates also PPFD 50, which is the light intensity which half saturates J. The fitting procedure is very similar to what we have seen before. The objective cell in solver is M4, which is the sum of the squared residuals, that is the distance between the function and the data point, and the variable cells are JSAT and theta. Theta is constrained to a value between 0 and 1. This procedure can be repeated with any of the three calibrations that can be specified here. For instance, by specifying Valentini, J is automatically recalculated. So after the function has been fitted, now J sat and theta will represent the Valentini calibration. It's interesting to note that when the specified calibration is changed, automatically the initial slope is transferred from the relevant sheet. When Bellasio is specified here, it is required to specify what YJLL is desired. It is suggested to specify the Yin initial quantum yield for electron transport, because the Bellasio calibration is a variant of the Yin calibration. When this is not available, one can specify V here. Sheet 10 calculates the rate of Rubisco oxygenation, the rate of Rubisco carboxylation, and the photorespiratory CO2 release. The data used here are the light curve and the ACI curve measured under ambient oxygen, because under low oxygen, photorespiration is assumed 0. The sheet allows to select what calibration to use to calculate J, which is the fundamental quantity on which VO, VC, and F are calculated. So, alongside the 3 calibrations already described in sheet A, here we can use also the fitted values for J, which were derived in sheet 8-9. These are only available for the light curve, so when F is specified in cell J2, the ACI curve J cannot be calculated. Similarly to what we have seen before, here we can specify what respiration in the light to use. The values are automatically calculated. Sheet 11 calculates the Rubisco CO2-O2 specificity factor SCO. This procedure is an upgrade of the original method presented by Yin. The data used here are the initial part of the ACI curves measured under ambient and low oxygen, carboxylating efficiency measured under ambient oxygen, and carboxylating efficiency measured under low oxygen, derived in sheet 5A and 5B, are also used. This sheet allows to choose what respiration in the light to use. This can be a number or one of the values derived in sheet 2. Similarly to what we have seen before, residuals can be log transformed. The sheet calculates a modeled assimilation, which is based on carboxylating efficiency and on assimilation measure under low oxygen. Then, by minimizing the difference between modeled assimilation under ambient oxygen and measured assimilation under ambient oxygen, SCO is derived. Practically, solver is used. So the objective cell is the sum of the squared residuals, which is the distance between modeled and measured assimilation under ambient oxygen. This is minimized by changing the value of SCO. This SCO value is automatically transferred to all following sheets. However, it is better to derive SCO on a number of healthy plants and then average. Sheet 12 estimates mesophyll conductance to CO2 diffusion. Briefly, it works on the principle that on the basis of PPFD and yield of photosystem 2, the rate of electron transport J is calculated. Then, on the basis of Ci and J, assimilation is modeled using the electron transport limited equations of C3 photosynthesis. Modeled assimilation is plotted in the graph as crosses. Mesophyll conductance to CO2 diffusion is estimated by curve fitting, that is, by minimizing the difference between the crosses and the dots, which is measured net assimilation. Here, there are two groups of data points. The first group of data points is the electron transport limited part of the light curve. That is, those data points which were assigned limitation 1 and 2. The second group of data points are the electron transport limited data points of the ACI curve. That is, those data points which were assigned the limitation 3. These are plotted together, however, only the electron transport limited part of the ACI curve can be selected, and this is done by changing the input in cell I3. So if we say no, just the ACI curve is plotted. If we say yes, both the light and the ACI curves are plotted. Secondly, it can be specified, as we have seen already, what calibration to use to calculate J. This can be the Yin, the Valentini, or the Bellasio calibration. Then we can specify as usual whether to log transform residuals and which respiration to use. Here in Q3 we can specify what Rubisco specificity factor to use. By default, this is copied from sheet 11, but if preferred, another value can be used by just typing the number in here. For instance, 2400. The value of GM, which minimizes the sum of residuals, is found with solver. In solver, we specify the objective cell as Q7 by changing variable cells Q6 and GM can be constrained to physiological values greater than 0 and smaller than 1. The fitted value of GM appears in cell Q6. Sheet 13a models assimilation using the model of Farquhar. The model of Farquhar is a mechanistic model based on Rubisco kinetics. The particular formulation used in sheet 13a is that of Ethier and Livingston. This is a formulation based on Ci, therefore there is a finite mesophyll conductance, and on gamma, the experimental compensation point. Gamma is copied from sheet 5a, whereas GM is copied from sheet 12. We will see later how these numbers can be overwritten and fitted concurrently. Because this model is based on Rubisco kinetic, it is valid only when assimilation is limited by Rubisco activity. Therefore, the data points copied in this sheet are those which were assigned Rubisco limitation, the T1 and 2. Different methodological alternatives are possible. In particular, different parameters can be fitted or assigned a literature value. For example, we could fit both the apparent Rubisco Michaelis-Menten constant and Vc max together in a single fitting step. In alternative, Km could be set to a literature value, for instance 550, and only Vc max can be fitted. We remove Km from the variable cells and we fit only Vc max. Other methodological alternatives can be fitting the compensation point together with the rest of the curve. So to do so, the compensation point can be added here in the variable cells. So this compensation point is now fitted together with VC max. To restore the link, I can just type equals gamma ambient. Another possible alternative is to fit GM together with the VC max. So in this case the procedure is exactly the same. GM is just added in the variable cells here. So now we have a fitted value of GM, a fitted value of VC max, and the literature value for the apparent Michaelis-Menten constant. The link with sheet 12 can be restored by just typing equals GM. Sheet 13b is similar to sheet 13a, just the data copied here come from the ACI curve measured under low oxygen. Similarly, the compensation point is that fitted under low oxygen, and respiration in the light is also the value found under low oxygen. The procedure is exactly the same. For instance, VC max and Km can be fitted together. So these are now the values of VC max and the apparent Km which fit the curve. We have now gotten to the end of this first video with the general features. I will upload another video with the advanced and additional features. Thank you for watching and enjoy hassle-free data analysis with my Excel fitting tool.